this session we are just continuing with the post-fertilization event. In our previous classes we already covered the endosperm, and today we just going to discuss about the development of dicot embryo. So today's topic is development of dicot embryo. So in fertilization we all know that there is a embryo sac, and after first fusion of your male gamete to the egg in the egg apparatus, the first cell which is formed, that means the egg is turned towards, that means the formation of your egg takes place, and after the first fertilization the zygote is formed. That means the zygote is gives rise to embryo, that means the zygote, from the zygote the embryo is formed. In the other hand, the nucleus is, that means the two polar nuclei fuses with the another male gamete, and from the another male gamete the triple fusion is takes place, and it result to your endosperm. So the total technique is always known as a double fertilization. All these things are we already discussed. So the first embryo is formed from the zygote. So our ultimate starting point is your zygote. So first start with your zygote. So here is your zygote, and the place of your zygote is, here is your microphylla end, another one is your salazar end. Nextly, the microphylla end takes a mitotic division. That means it takes a mitotic division. A mitotic division takes in your zygote. That means the zygote is going to split through mitotic division, and it gives rise to two cells. That means the zygote is going to divide through mitotic division, and two cells are formed. And the parts are also same. One is your microphylla, another one is your salazar, and the cell towards your salazar end is organically known as your terminal cell. So this is your terminal cell, and the cell which is towards your microphylla end organically known as your basal cell. So in some books you saw that they mentioned that the cell towards your upper side is known as your terminal cell, and the cell towards your lower side is always known as your basal cell. But in every cases it is not equal. That means it creates a confusion to all of you. Just take an example over here. If we talk about the most common type of ovule, that means your anatopous ovule, it is your microphylla end, it is your salazar end, because it is your offside down structure. So here is your mouth part, so it is your microphylla end, it is your salazar end. In this case, they told that the upper part is your terminal end and the lower part is your basal end. It is true. But if we take an orthotropous ovule, that means if we take this type of ovule, I just roughly draw a diagram over here, and here is the zygote. When the mitotic division takes place, it is the microphylla end, obviously, it is the salazar end. So the cell towards, that means the upper cell is your terminal cell according to them, and the lower cell is your basal cell. I just wrote an abbreviation, BC, basal cell. So this is your PC, terminal cell, this is your BC. That means according to them it is totally opposite. So whenever you draw this type of diagram, just mention the salazar end and microphylla end. Just points where it is arise, the place. So to avoid this confusion, just wrote like this. That means the cell which is towards your chalazal end is known as your terminal cell. The cell which is towards your micropylar end is known as your basal cell. Next, we just change the color. So in basal cell, that means the cell towards your micropylar end, there is a transverse division takes place. So what happens over here? There is a transverse division takes place. By this transverse division, there is a thick, that means it look like a stalk of wine. This structure is formed. 7 to 8 cells of suspensor are formed. These are otherwise known as your suspensor cells. Draw it a little bit bigger. So these types of suspensor cells are formed, and nextly a haustorial cell is also formed. This is your haustorial cell. And this is your suspensor cell. So what happens? I again repeat. From the basal cell, it forms a transverse division, and the cells is divided to form 7 to 8 number of cells, which is already known as your suspensor cell, and the lower cell is already known as your haustorial cell. Next, the terminal cell is also changes, but in terminal cell, the division takes place in all the planes. That means it gives rise to form a mass of cells. So when the terminal cell is going to divide, that means the cell towards your chalazal end, this is going to divide. It is dividing transverse division. It is divided in all planes. And what is formed? It forms a mass of cells over here. And at first, 2 divisions are takes place and 4 cells are actually formed, and this condition is always known as your proembryo. That means the condition which is formed from the terminal cell to the proembryo in all planes are known as, that is, it is known as your proembryo. In some books you found that it is written as proembryonal quadrant, because as it is divided to form your 4 cells, so it is written as a proembryonal quadrant. Next key happens. See, nextly, the proembryonal quadrant again divide, and it forms 8 cells. So we just draw the same diagram. It looks like a ball shape. I again told you that the total structure, total cells are not accurately look like that. We just draw the cells. For a better understand, we just draw every cells and we just wrote the fate of the cells. Actually, these cells are not like that. I told you that it is a mass of cells. That means it look like a ball. If you take an example of a ball and try to see all the planes, it is not visible to you. That means the cells which is in front of you, it is visible to you, but the back side of the cells are not visible. Just to look all these cells, we just draw these types of diagrams. So what happens over here? The cells are going to divide. So what happens? A ball of cells are formed That means 8 cells are formed. 1, 2, 3, 4, 5, 6, 7 and 8. That means these 8 cells are formed, and this structure is organized known as it is your proembryo. This is also your proembryo, and it is known as your proembryonal octant. As there is 4 cells, then it is known as your proembryonal quadrant. Here 8 cells, so it is known as your proembryonal octant. Nextly, what happens? The first cell, which is placed over here, the first cell of suspensor which is attached to your proembryo is changes. That means this cell is changes. I just change the color. This cell is changed, which is attached to your proembryo, and this cell is otherwise known as your hypophysis. So this is your hypophysis. Next, what happens? All the cells are loop cell. That means a single cell of suspensor is changed, and all the cells are as it is. So here all the cells. And the structure of osteoid cell. So the suspensor is as it is, and the osteoid cell is also as it is, and a single change takes place in your hypophysis. In the next stage, what happens? The next stage is the formation of your dermatocyte. That means a periclinal division is takes place in your proembryo. So we just continue with this class. So here is your proembryonal octant. That means your proembryo. We just draw first the 8 cells. That means this is your 8 cells. Nextly, there is a periclinal division takes place. That means it's divided such a manner that one cell is towards your outer side and one is your inner side. So here is your outer side, inner side, outer side, inner side, outer side, inner side, outer side, inner, and here is your outer, inner. So a layer of cell is takes place, and the cells are otherwise known as, that means the layer is otherwise known as your dermatogen. So this dermatogen again it forms the epidermis. So what happens over here? The proembryonal octant is again changes to form a dermatogen, because the division is takes place periclinally, and here I wrote that a division is takes place here, periclinal division. So what happens here? The periclinal division takes place and a dermatogen takes place. Later the dermatogen forms your epidermis. Next, the cell towards your hypophysis is going to divide and form 8 cells. We just divide like that, 6 and 8. So 8 cells are formed. Nextly, these 8 cells gives rise to the structures. We just study how the structures are formed, and the suspensor is as it is. So if you look the diagram in comparatively manner, the suspensor is look bigger over here, but where the proembryonal octant and dermatogens are developed, in that conditions the suspensors are comparatively look smaller. And later some couple of stages I told you what is the work of your suspensor cell actually. So this is your suspensor. And the host cell as it is. Nextly, the formation of your epidermis takes place. Next, we just cover a stage which is always known as your globular stage. So it is your globular stage. As I told you that it is the fate of cells. That means the total structure is not look like that. So we just naming these structures, how they are formed. That means which part forms the endosperm is gives rise to what? That means in plant structure of a baby plant, what type of cells are going to divide? That means the cells which are formed over the epidermis, that means in your epidermis, in your suspensor cells or in your posterior cells, what they are going to form. In your first, the 4 cells, that means these 4 cells, which is towards your suspensor cell or towards your micropara cell, is formed the root tip or root cap. That means these are formed root tip and root cap. That means they form the radical part. Nextly, the upper 4 cells which is attached to your octant structure, that means your proembryo, is form your root cortex. I again repeat, the cell towards your micropara or the cell is attached to your suspensor form your root cap or root tip, that means your radical part. Nextly, the cell which is attached to your proembryo forms your root cortex, that means these 4 cells form your root cortex and the lower 4 are form your root tip or root cap. Nextly, in proembryonal octant, the lower 4 cells, that means 1, 2, 3 and 4, these 4 cells formed, I just erase it a little bit. So the cells which is towards your lower, that means you attach to your root cortex cells, they form the hypocutae. That means the lower 4 cells form the hypocutae and the upper 4 cells form the plumule or the cotyledon. Both plumule and cotyledon. That means what type of changes are takes place? The dermatogen forms your epidermis. The total structure, in total structure, 4 cells which is towards your micropara or which cells which are attached to your suspensor form your root tip or root cap. Next is the cells which is attached to your proembryo forms your root cortex. Nextly, in your proembryo, lower 4 cells form your hypocutae and the upper 4 cells form your plumule or cotyledon. I hope this is clear to all of you. Nextly, why we draw this type of cells? We just draw this type of cells because of these names. Because of these cells, we know that how the root tip is formed, how the root cap is formed, which part is formed your radical, which part is formed your cotyledon, which part is formed your plumule, all these things. So this is one structure of your globular cells. It is just the one step of your globular cells. All the other stages are also there. It is just the globular cells. Nextly, the work of your suspensor cell. If we look at these cells, that means looks like a stalk of coil-like structure. If we draw the most common type of ovule, which which we already covered in our previous some classes. So if you draw the ovule, it somehow look like that, and here is your embryo, that means your egg apparatus or your embryo sac, and here is the formation of your zygote takes place, and nextly zygote is divided. I just roughly draw this diagram, don't judge it actually. So what happens here? Here is the zygote. I told you that here is the zygote. Nextly mitotic division takes place. Nextly the globular stage takes place. That means the proembryo is formed. We just changes the structure. After that means we also know that the endosperm is already formed. That means it is filled with the endosperm. That means the nutritive tissue. As I told you that everything that happens in nature, that happens for a purpose. That means at the time of formation of this zygote, its food is already ready. That means the endosperm is already formed. So there is the zygote. So at the time of ready of the zygote, endosperm is also ready. Nextly, what is the cause of your suspension cell? If we change the diagram, it is the stage of your mitotic division. After the mitotic division, we just change it to the third stage. In third stage, what happens? The development of your suspension takes place. If we draw a globular structure, nextly the suspension cells and the host. If we draw this type of diagram, we look at the suspensor is push the proembryo, that means the embryo, towards the endosperm, that means towards the nutritive tissue to the nourishment. So the main function of your suspensor cell is to give nourishment. That means by this suspensor cell, it just push up the proembryo deep into the endosperm, and it can receive the nourishment, that means the food material, and grow. So this is the main function. That means here is your endosperm, and it is just push the proembryo. So it is the main function of your suspensor cell, and we just continue these terms towards your globular cell stage. In our next class, we just continue other stages. That means in later on, we just continue the globular stage and how the changes are taking place. This is the globular cell. Nextly, the hot cell and other structures are going to form. So hope this class is helpful to you. Thank you.